with your system, not by tweaking or rewiring the hardware, but actually you can take a step back and give it stimuli or inputs the way that you would give to a reprogrammable computer, and cause the cellular network to do something completely different than it would otherwise have done. So the ability to see these bioelectrical signals is giving us an entry point directly into the software that guides large-scale anatomy, which is a very different approach to medicine than to rewiring specific pathways inside of every cell.
And so in many ways, this is the amazingness of your work, is that you're starting to crack the code of these electrical signals. And you've got an amazing demonstration of this in these flatworms. Tell us what's going on here.
So this is a creature known as a planarian. They're flatworms. They're actually quite a complex creature. They have a true brain, lots of different organs, and so on. And the amazing thing about these planaria is that they are highly, highly regenerative. So if you cut it into pieces, in fact, over 200 pieces, every piece will rebuild exactly what's needed to make a perfect little worm. So think about that. This is a system where every single piece knows exactly what a correct planarian looks like and builds the right organs in the right places, and then stops. And that's one of the most amazing things about regeneration. So what we discovered is that if you cut it into 3 pieces and amputate the head and the tail, and you just take this middle fragment, which is what you see here, amazingly, there is an electrical gradient head to tail that's generated that tells the piece where the heads and the tails go, and in fact, how many heads or tails you're supposed to have. So what we learned to do is to manipulate this electrical gradient. And the important thing is that we don't apply electricity. What we do instead was we turn on and off the little transistors. They're actually ion channel proteins that every cell natively uses to set up this electrical state. And so now we have ways to turn them on and off. And when you do this, one of the things you can do is you can shift that circuit to a state that says, no, build 2 heads, or in fact, build no heads. And what you're seeing here are real worms that have either 2 or no heads that result from this, because that electrical map is what the cells are using to decide what to do. And so what you're seeing here are live 2-headed worms. And having generated these, we did a completely crazy experiment. You take one of these 2-headed worms and you chop off both heads and you leave just a normal middle fragment. Now keep in mind, these animals have not been genomically edited. There's absolutely nothing different about their genomes. Their genomic sequence is completely wild type. So you amputate the heads, you've got a nice normal fragment, and then you ask in plain water, what is it going to do? And of course, the standard paradigm would say, well, if you've gotten rid of this ectopic extra tissue, the genome is not edited, so it should make a perfectly normal worm. And the amazing thing is that that is not what happens. These worms, when cut again and again in the future in plain water, they continue to regenerate as 2-headed. Think about this. The pattern memory to which these animals will regenerate after damage has been permanently rewritten. And in fact, we can now write it back and send them back to being 1-headed without any genomic editing. So this right here is telling you that the information structure that tells these worms how many heads they're supposed to have is not directly in the genome. It is in this additional bioelectric layer. Probably many other things are as well. And we now have the ability to rewrite it. And that, of course, is the key definition of memory. It has to be stable, long-term stable, and it has to be rewritable. And we're now beginning to crack this morphogenetic code to ask, how is it that these tissues store a map of what to do, and how we can go in and rewrite that map to new outcomes.
I mean, that seems incredibly compelling evidence that there's, that DNA is just not controlling the actual final shape of these organisms. So there's this whole other thing going on. And boy, if you could crack that code, what else could that lead to? By the way, just look at these worms. What is life like for a 2-headed flatworm? I mean, it seems like it's a kind of a trade-off. Like, the good news is you have this amazing 3-dimensional view of the world, but the bad news is you have to poop through both of your mouths.
So the worms have these little tubes called pharynxes, and the tubes are sort of in the middle of the body, and they excrete through that. These animals are perfectly viable. They're completely happy, I think. The problem, however, is that the 2 heads don't cooperate all that well, and so they don't really eat very well. But if you manage to feed them by hand, they will go on forever. And in fact, you should know these worms are basically immortal. So these worms, because they are so highly regenerative, they have no age limit. And they're telling us that if we crack the secret of regeneration, which is not only growing new cells, but knowing when to stop. You see, this is absolutely crucial. If you can continue to exert this really profound control over the three-dimensional structures that the cells are working towards, you could defeat aging as well as traumatic injury and things like this. So one thing to keep in mind is that this ability to rewrite the large-scale anatomical structure of the body is not just some weird planarian trick. It's not just something that works in flatworms. So what you're seeing here is a tadpole with an eye and a gut. And what we've done is we've turned on very specific